Fantastic. Well, here we go. It gives me great pleasure to introduce our next speaker. Tom Booth is a postdoctorate researcher at the Natural History Museum and has a big interest in ancient DNA, various other things like taphonomy and funerary arrangements as well. But today he's going to be talking to us about Cheddar Man and revealing some of the secrets that the ancient DNA of Cheddar Man has told us about the people of Britain. So please give a warm welcome to Tom Booth.
Thanks for that, and thanks very much for coming, everyone. Can everyone hear me okay? Yep. Okay. Okay, so this talk is basically going to condense what we've learned through the last 10 years of ancient DNA studies, mostly looking at prehistoric human remains, so DNA extracted from prehistoric human remains from Britain, dating to the last 10,000 years or so, and what we've learned about the population history of Britain and Europe more broadly, as well as specifically about this individual, Cheddar Man, who, as many of you will be aware, hit the headlines about a year ago for various reasons. So focusing on Cheddar Man to begin with, so Cheddar Man is a skeleton that was found in Goff's Cave in the Mendip Hills in Somerset, and it was originally found back in 1903, so the standards of excavation weren't necessarily the best, but they managed to retrieve what was essentially a complete skeleton, more or less, that you can see here, the newspaper reports at the time. And what was interesting is that even though this was, over 100 years ago that they found this skeleton, this is still the oldest complete skeleton that we have from Britain. So there are older human remains that we have from various places around Britain, but they tend to be bits and pieces of bits of bone, whereas Cheddar Man is the oldest that we have, where it's more or less a complete skeleton. So he's been radiocarbon dated twice, actually, and he's dated pretty securely to around 10,000 years ago, so 8,000 BC. Osteological assessment of his skeleton, so looking at the morphology of the bone, suggests he was male, 160 centimeters tall, died probably in his early 20s, and dates to this a period known as the Mesolithic, so that's the Middle Stone Age, and the specific period known as the early Mesolithic. So Britain at this stage, if you can imagine such a thing, was still attached to continental Europe by an extensive lowland landscape known as Doggerland. So essentially, Britain at this point was kind of a northwestern peninsula of Europe, mainland Europe. And the people at this point were still hunting and gathering using stone, bone, and antler tools. Probably quite mobile, maybe mostly living in temporary shelters, eating things like wild aurochs, fish, wild pig, wild deer, and gathering things like hazelnuts, and using this specific I can't use that. The specific tools that you can see in the top right-hand corner, known as microliths. And even though they're one of the best sites to look at in terms of to understand Mesolithic lifeways, and this is Starcar in North Yorkshire, which is this big Mesolithic sort of settlement, but also ritual complex where people, they found these antler headdresses that were made out of red deer frontlets, so the front part of the skull and the antlers. So these people were, just as sophisticated as we are today, culturally, cognitively, and they had rich ritual lives, despite being quite mobile. So even though Cheddar Man was found in a cave, there's no evidence that people were living in caves during this time. They weren't being occupied. They were probably living in sometimes wooden houses or temporary shelters made of skins. And the majority of human remains that we find dating to the Mesolithic period, this early Mesolithic period, come from caves. So it's likely that he was put in the cave as part of a funerary, an established funerary tradition. So moving on to the ancient DNA, just to go through the advancement in the last 10 years or so. So Up until around 10 years ago, analysis of DNA from ancient remains was based on this technique, polymerase chain reaction, where what you were doing was essentially fishing out predefined bits of DNA from these ancient remains. So usually you were looking for particularly informative bits of the DNA. So things like the mitochondrial DNA to look at maternal ancestral markers, or the Y chromosomes to look at paternal ancestral markers, or looking at parts of the genome or genetic variants that we know are linked to functional things, so things controlling whether you can digest milk, that kind of thing. And this typically involved looking at DNA sequences, so those sequences of nucleotides that are denoted by GCTNA, usually looking at only hundreds of thousands, sequences of about hundreds to thousands of these nucleotides, base pairs. What happened around 10 years ago was the next generation sequencing technology came around, and what this allows you to do is that rather than fishing out small informative bits from the genome, you essentially put catalog DNA strands in the whole ancient DNA sample, and essentially just get a random distribution of DNA sequences from within that sample. So a sample of more or less all of the DNA that's in there. So you get essentially an individual's whole genome. So genetic information from all 23 pairs of chromosomes and things like the mitochondrial DNA. So you're looking at, rather than hundreds to thousands, you're looking at hundreds of thousands, potentially millions of base pairs from ancient individuals. So this gives us the ability to look at a much richer idea of people's ancestry, rather than, for instance, looking at the specific paternal or maternal ancestry. You can look at an individual's entire ancestry, and then you can make inferences about their populations. What's also helped is that one of the biggest problems with this technology is that the majority of DNA that you get in an ancient sample actually comes from bacteria that have colonized that human-born since that individual died. But fortunately, what we have now is this bone called the petrous portion of the temple bone that we routinely sample, because it routinely preserves DNA incredibly well and keeps out the kind of bacteria and things, chemicals that could break down the DNA within bone. So the petrous portion of the temporal bone is this bone on the side of your head here around your ear, and the petrous portion is actually surrounds your internal ear canal. So it's an incredibly dense piece of bone. In fact, it's the densest bone in the human body, which is why it preserves the DNA so well. And so by sampling the petrous bone, we were able to go from a situation where we were getting sort of 2 or 3% of a bone sample came from an ancient human to 40, 50, 60, 70, sometimes 80% of the DNA that we're getting out of a bone sample came from an ancient individual. So this is being able to sample this element has made the process much more efficient, and we get a much richer level of data from things. So probably many of you are old enough to remember the original study of Cheddar Man that was done, I think, back around 1997 by Brian Sykes at Oxford University. And what Brian Sykes did was use this early method to try and look at the mitochondrial DNA of Cheddar Man. He drilled into Cheddar Man's tooth and attempted to extract the mitochondrial DNA to look at his maternal ancestry. And he found that his results suggested that Cheddar Man belonged to this mitochondrial haplogroup, so this particular type of maternally inherited DNA that's classified as U. And this was reported at the time as suggesting that, so they found this, they basically also did the mitochondrial sequences, the maternal DNA of lots of people who lived in the local village, Cheddar Village, and looked to see what their mitochondrial sequences was. And they found this guy, Adrian Target, who was a local school teacher, who also had the mitochondrial haplogroup of U. And this was used, particularly in the papers, to suggest that there had been continuity in Cheddar Gorge in population for the last 10,000 years, and that Adrian Target was a direct descendant of Cheddar Man. So the problem with this analysis, to some extent, I mean, he did the best that it could with the methods that were available in the 90s, but the problem is that the methods that were used by then, the PCR methods, it's very difficult to be able to control for contamination. So it could be that he had a genuine result, the team at Oxford had a genuine result, but it's impossible to be able to test that. It's very difficult to be able to test that, but you can test the authenticity of ancient DNA sequences now through various methods. The also problem was the way that this was reported misunderstood what mitochondrial DNA can be used to say, in that it's vaguely possible that Adrian Target was a direct maternal ancestor of of Cheddar Man, but much more likely is that Cheddar Man and Adrian Target shared a maternal ancestor tens of thousand years in the past. So it didn't really say anything about continuity of population in this particular area. So we've, at the Natural History Museum, we looked at Cheddar Man's genome as part of a broader study looking at population history in Britain through prehistory, particularly at the transition from the Mesolithic, which is the period to which Cheddar Man belonged, and the Neolithic, where you get the introduction of farming practices to Britain. And to understand the genetic pattern that we see in Britain, we need to understand what's happening in Europe. So this starts off a lot. From before the last glacial maximum, so before about 20,000 years ago, there's more to the story, but it's a lot more complicated. So I'm going to start after the last glacial maximum. So from around 18,000 BC, so 20,000 years ago, Europe seems to be reoccupied by this population, hunter-gatherer population, that moves from southwestern, and this is based on ancient DNA extracted from ancient remains all around Europe that you can see on here. So after around 20,000 years ago, Europe is reoccupied. The glaciers retreat, and Europe is reoccupied by this population that seems to move out from the southwest, so probably southwest Spain, referred to as the El Miron cluster, after the earlier site from which the first person who belongs to this cluster belonged. But then what happened, so this is represented by these green dots you can see here. But starting from around 14,000 years ago, so about 12,000 BC, the genetics of people living in Europe substantially changes, suggesting that there is an almost complete replacement of that earlier population by another hunter-gatherer population who expands out of, we're not sure exactly where, but somewhere in the southeast, maybe Southeast Europe or perhaps the Near East. So we get a complete population change there, which is demarked by the red dots. So that's going through time. You get the green dots, and then you get the red dots. So the green ancestry is replaced by the red ancestry. So when thinking about the British Mesolithic, the question is whether or not 10,000 years ago, whether or not Cheddar Man and other individuals from the same period are associated with that earlier population and persisted from the end of the Ice Age into the Mesolithic, so into the Holocene, or whether they may have belonged to this new population that expanded from the southeast, and that this population essentially came across and swept all the way up into northwest Europe. Also, what happens is another population movement that occurs seemingly specifically in Scandinavia, where this group that's moved in what are called the Villa Brunio or Western Hunter-Gatherer cluster are met by another group who are from Russia, what is now Russia, expanding into Scandinavia. And it's important, and mixing. So you get this very particular population who are living in Scandinavia, who are this admixed population between these people who expanded in from the southeastern Europe and these people that are coming from parts of what is now Russia. And it's important to emphasize the diversity in these populations that are mixing. So because these populations have essentially been isolated from one another for potentially tens of thousands of years, they're very genetically differentiated from one another. So the genetic difference between these mixing populations is sort of on a continental scale when we look at modern populations today. So the difference between these two populations mixing is this difference between typical individuals living on different continents today. That's how diverse these two are. So in Scandinavia, you have this particularly this particular population as well, these Scandinavian hunter-gatherers. So it's possible that Britain could have been influenced by this population via Doggerland. So this is what's called a principal components analysis, a PCA. So I'm going to have to talk you through this rather than being able to show you. So this is essentially a map of genetic difference and similarity of different populations. So in the middle, the kind of slightly faded markers are all modern populations living in Europe, whereas the darker or the more solid markers are ancient individuals that have been compared. So you can see this essentially showing how similar or different ancient and modern individuals are. So this population that is called the Western hunter-gatherers, who are occupying Europe in the Mesolithic, are the burgundy symbols that you can see to the left of this plot. The individuals dating to the British Mesolithic, including Cheddar Man, are the green the green plots that you can see there. So essentially, as you would expect, that Cheddar Man and other individuals occupying Europe at during the Mesolithic look genetically similar to other Mesolithic Europeans, and that this population movement of people out of southeastern Europe does extend all the way up into Britain, and there's no remnant of that earlier population that had expanded into Europe after the last glacial maximum. What's interesting to note about this Mesolithic population is that it exists entirely outside of the variation that you see in genetic variation that you see in modern day Europe. So they are a population that essentially genetically doesn't exist anymore. They are kind of almost alien in that they are completely outside of modern European genetic variation. So one of the things that was looked at originally in some of the early people belonging to this population were genetic variants that we know are associated with skin pigmentation in modern Europeans. So there are two particular variants that explain a lot of the kind of paler skin pigmentation that you see in modern Northern European populations. So some of the early studies looked at these variants to see when this comes along, because they were interested because the evolution of paler skin is interesting because it's thought to be as a result of an adaptation to living in more northerly latitudes, where you have less sunlight and your body absorbs UV and sunlight in order to synthesize vitamin D. So if you have lighter skin is more efficient at doing this. So if you're living in more northerly latitudes, then you would adapt to have paler skin in order to be able to absorb UV light to synthesize vitamin D, and so you're not vitamin D deficient. But when they looked in this particular Western hunter-gatherer from Spain, they found that he had neither of these variants of a gene linked to skin pigmentation that is associated with lighter skin pigmentation in Northern Europeans, which would predict that he would have likely had had dark skin. But interestingly, he did have the genetic variant that is most commonly associated with blue eyes. So you had this interesting, and he had other genetic variants associated with darker hair. So it's likely they would have had this strange combination of traits of dark skin, dark hair, but blue eyes, or weird combination that we consider today in Europe, suggesting that the common genetic variants that are associated with, the sort of what is regarded as with the typical European phenotype of blonde hair, blue eyes, pale skin, all came into Europe quite separately from one another, and then merged in Europe as a result of different populations moving in with these traits. So what we did was, at the time when we were doing this analysis, Channel 4 were doing a documentary where they wanted to do a facial reconstruction of Cheddar Man, and they wanted to include the ancient DNA analysis in the reconstruction in order to understand what his physical characteristics might have been. So there had just been this forensic method developed in the US, which looking at modern populations, looked at genetic variants associated with skin pigmentation in modern populations and used that and sort of classifying modern people's skin pigmentation using this Fitzpatrick scale, so quite crudely, but in kind of this categorical way, in order to predict people's skin pigmentation for forensic purposes. So we applied this method to the variants associated with Cheddar Man. So looking rather than at just these 2 variants that had been looked at previously, we looked at 36 genetic variants associated with skin pigmentation. And the prediction that came back when we did that was that he would have had sort of somewhere between these last 2 categories, so brown to black, what is described in the prediction model as dark to dark black skin. Again, dark brown hair, probably blue-green eyes. So what this suggests naively is that Cheddar Man probably had quite dark skin pigmentation. And that's still, at the moment, the best prediction that we have for him. What it could be, alternatively, is that because Cheddar Man belongs to this population that essentially doesn't exist anymore, that is entirely, mostly unrelated to modern populations who currently inhabit Europe, that he had genetic variants linked to skin pigmentation, that a lighter skin pigmentation or any other skin pigmentation, that essentially don't exist anymore, that we haven't found yet because no population currently carries them. So to me, anyway, both of those results mean the same thing, or at least the other one's weirder than the other. Either he had dark skin, or his genetic architecture of skin pigmentation is something that doesn't exist in modern populations anymore, which reflects the fact that he is from this population that similarly doesn't really exist anymore and is completely detached from the current inhabitants of Europe and Britain more generally. If you're interested in this kind of thing, so we also looked at his Y chromosome and mitochondrial haplogroup. So it belongs to, and this will be important later, the I2A2 Y chromosome haplogroup and the U5B1 mitochondrial haplogroup, which is interesting. The U5 result was interesting because it's similar to the one that Brian Sykes got, but it's difficult to say whether, because of the potential contamination issue, there are enough people around with a U in Britain with a U haplogroup that it's plausible that just by coincidence someone with that U haplogroup could have contaminated the Cheddar Man sample that was taken originally. So it's interesting that it verifies that result, but it's the same as that result, but we don't know whether that means that back in the 90s they got the correct result or not. It's impossible to say at the moment. So in terms of thinking back to old Adrian Target and the kind of genetic legacy of Cheddar Man today, so this is a picture of Charlemagne. Now I've included this because there's always the idea about Charlemagne, people being descended from Charlemagne, and legends about people being descended from Charlemagne. And if any of you are familiar with Adam Rutherford's work, who deals with a lot of this stuff about ancestry, essentially Cheddar Man existed so far back in time that he is, if he has any descendants at all that exist today, basically is the ancestor of everybody alive on Earth today, more or less, because it existed so far back in the past. So the reason why I included Charlemagne is that they've calculated this identical ancestor point for people with recent European ancestry. So this is the point at which everybody that existed in a particular place is the ancestor of everybody alive today in that particular place. So for people with European ancestry, everybody who was alive in Europe in the 10th century is their AD is their ancestor. It's the identical ancestors point. So in the 10th century, if you have recent white European ancestry, everybody who existed in Europe probably is your ancestor. There is no specificity in your ancestry, and probably much wider afield as well. So when you get to 10,000 years ago, it's more or less the entire world. Yeah, including Cheddar Man, if he indeed passed on any descendants, which he may not have done. So moving on from, so why why does this population essentially not really exist anymore? So moving on from Cheddar Man into and the Mesolithic into the Neolithic period in Europe, and the Bronze Age to some extent, essentially population history of European population prehistory of Europe is defined by these episodic large-scale transformations of genetics of populations, suggesting the influence of migrations of people, again, that are very different from the populations that are inhabiting these areas previously. Starting at around 6,000 BC, so about 8,000 years ago, you get the movement of the emergence and movement of farming cultures across Europe. And that movement of farming cultures and that development of farming cultures from the ancient DNA that has been looked at is associated with this ancestry, this new ancestry originating around the Aegean. So these populations move in two different directions across the Mediterranean and northwest into Central Europe, and they kind of mix with the local populations as they go. So as they move further west, they incorporate ancestry from the Western hunter-gatherers, the Mesolithic populations that were there before, but this ancestry is always a minority of these populations that are moving, presumably because what farming allows you to do is to maintain a much larger population size than if you're hunting and gathering. So, these two populations could be, and likely are to some extent, mixing completely eventually, but the Mesolithic hunter-gatherers have a much lower genetic legacy because they have a lower population size. Then what happens from around 4,700 years ago, so 2,700 BC, in Northern Europe specifically, the genetics of the population transform completely again with a very divergent population seeming to influence the genetics of Northern and Central Europe. Again, this population is on a continental scale different from the population that was here before. And this seems to suggest migrations of people from the Pontic steppes, so just north of the Black Sea, into these areas of Europe. And again, you get mixing with the populations that were already there, but that this, what was referred to as steppe ancestry, tends to predominate in these populations, despite them being quite mixed. So what's the story in Britain? How does it fit in? Britain's an island by this point. It becomes an island around 8,000 years ago, so it's still in the Mesolithic. And from around 4,000 BC, so 6,000 years ago, you start to get the appearance of farming cultures in Britain. So these are defined in Britain by these megalithic tombs, which you can see in the top left. This is the West Kennet tomb. The arrival of things like these ground stone axes. For the first time, you get pottery in Britain. So you get this carinated bowl pottery and these particular style of arrowheads, these leaf-shaped arrowheads. And then a millennium and a half later, I should add, because some of the research that we did has been very much linked to Stonehenge, but talking about Stonehenge being built by migrants, but Stonehenge isn't built until 1,500 years after these populations start to move into Britain. So if you've been in a place for 1,500 years and you're still being called a migrant, then your test for migration status is rather strict. I don't know, there still must have been Norman migrants around somewhere. So it's very much linked to these early farming cultures. So what we see when these cultures start to arrive is that the ancestry of Britain transforms as it does elsewhere in Europe, suggesting that these populations who moved across Europe over a couple of thousand years eventually make their way up into Britain. And the farmers in Britain are mostly associated with these people who took the Mediterranean route through Europe. So they moved down the Mediterranean over 2,000 years, up through into northern France, and then from northern France into Britain. But what's interesting about Britain is that, unlike in other areas of Europe, we don't see much mixing with the local Mesolithic population. So there is a detectable amount in Western Scotland, but outside of Western Scotland, we don't see any. And we don't really know why this is. I mean, the easiest explanation is that, for whatever reason, by the end of the Mesolithic in Britain, population density in Britain just wasn't all that high, and that subsequently, because the populations of farmers that were coming over were much larger than these hunter-gatherers, that the hunter-gatherers left little genetic legacy. So going back to that question about the legacy of Cheddar Man specifically, it's perhaps less likely that he passed on specific descendants. So in terms of skin pigmentation and the variants that we see in these populations, so these populations that move out of the Aegean carry one of those variants that is associated with paler skin pigmentation, or explains some of the variation in paler skin pigmentation in Northern Europeans, but not the other. So this will, if you put this through a kind of predictive model, this will predict that they would have variably had sort of skin pigmentation populations that we think about occupying regions around the Mediterranean, sort of in sort of the Mediterranean, the Near East and North Africa. And this individual this facial reconstruction was based on a skeleton found at the White Hawk Causeway enclosure, which is in Brighton. And we didn't get any DNA from this person, but this reconstruction of the skin pigmentation is based on a prediction of skin pigmentation and eye colour and hair colour in the British Neolithic individual from whom we got the most data from, so who the data is most reliable. So these populations seem to have had dark brown hair, predominantly but not exclusively brown eyes, and then variable, I should stress, variable skin pigmentation, but sort of more what we would associate with the populations that currently live around the Mediterranean. And then in Britain, what you get starting from around 2500 BC is the arrival of the Beaker cultural phenomenon, and this is a phenomenon that seems to begin in Iberia and spread all through a lot of Europe and some of North Africa and into Central Europe particularly. And it's quite a nebulous thing in order to get hold of, which is why it's called the phenomenon, in that there are certain traits that these different cultural groups have in common, there's certain that they don't, but broadly it's associated with this Beaker pottery, and with this Beaker style pottery, and a particular type of new funerary treatment, and a new type of kind of archery that you see, sort of an emphasis on archery that you see in some of the funerary environments. And this kind of culture is epitomised really in the British burial in Britain by the Amesbury archer, who has four of these Beaker pots, and lots of things to do with archery. I should stress that there's a lot of cultural variation in Britain and elsewhere associated with this, but there does seem to be a definite change with the arrival of this culture. And again, like in the Neolithic, there's always been a talk about whether the spread of this cultural package or this set of cultural traits is associated with a movement of people, or whether it was just a movement of ideas. So the analysis of ancient skeletons associated with Beaker cultures in Iberia and Central Europe suggests that these cultures spread from Iberia to Central Europe without any significant migration. So the genetic signature of Central Europe stays the same, and they just seem to adopt these Beaker practices. However, when these Beaker cultures first appear in Britain around 2500 BC, you see the genetics of the population completely change. They bring in this steppe ancestry that has been established in Central Europe, as well as the other ancestry that they've obtained from mixing with local populations in Europe. And that, over a space of around 200 years, it results in the almost complete replacement of the local Neolithic population. So around a 90% replacement. As you can see here, so this is just demonstrated in this figure that the blue represents the British Neolithic ancestry, and the red represents the continental Beaker ancestry. So you can see that after about 2450 BC, you get this complete change from blue to red. So again, the population completely seems to change and stays the same all the way through the Bronze Age, essentially all the way up until around 1000 BC, so 3000 years ago. And again, when you looked at pigmentation characteristics in these individuals, they seem to have more of the variants linked to lighter skin pigmentation as a whole in the population. Those variants still aren't as common as they are in Britain today. So, and there was still lots of variation in skin pigmentation. So these are two individuals where, again, we've used the same predictive models to indicate what they would have looked like. And you have one who's, like, keep thinking there should be a screen there. There's one that's blonde hair, blue-eyed, and blonde, but then there's another individual that we did this that would have had more kind of, again, this Mediterranean style, or what we consider today to be a Mediterranean style phenotype, with brown eyes, kind of olive skin pigmentation, and black hair. So again, there's still lots of phenotypic variation amongst these populations. So turning to the legacy, so in modern British individuals that have where documentary evidence suggests that they have recent ancestry in Britain, going back at least to their grandparents, we can look at the proportions of these three different populations. So the Western hunter-gatherers, the Neolithic farmers, and the steppe populations who are associated. This green population who are originally associated with Yamnaya cultures. We can look to see to what extent they are represented in contemporary populations living in Britain today. What's important to say is that the reason why we pick people who have a recent ancestry in Britain is to try and get as far back as possible, so we can kind of connect the dots and see what else, if there is anything else to find in terms of changes in ancestry and the people who are currently living in Britain. And what we find is that in England, and particularly southern England, that there is more of this Neolithic ancestry in southern England than there is in other parts of Britain, suggesting that there is another population change somewhere between somewhere in the Iron Age or Roman period that brings that substantially alters the genetics of at least southern English populations again. And this is kind of what we're looking for now, as to what is responsible for this. It's not the Saxons, so the Saxons do have an influence later on, particularly in the east of England, but that actually doesn't explain this. So what we're working on at the moment is figuring out what causes this genetic change, whether it is another movement, sudden movement of people, or whether it is something to do with the networks that are established in the Iron Age and the fact that you have kind of a constant flow of people in and out of Britain through the Iron Age. And whether it's associated with, migrations associated with Celtic cultures, Celtic languages, or possibly even whether the Romans have more of a genetic impact than has often been assumed. So going back to kind of the subtitle of my talk, talk about the origins of the British. I don't really know what this phrase means anymore, in a sense, in that if you're talking about the origins of the only consistent really definition of British are people who have lived in Britain, that the origins of Britain, is entirely dependent on what time period you're talking about, and that this has changed quite dramatically through time. And the thing is that you constantly have gene flow in and out of Britain, but the rate and the scale of the change sort of changes, and there are these episodic pulses of change, but it's always changing. So there's no static idea of what is a British person. And if you're talking about modern British people, then that's rooted. The explanation of people who are British today is rooted in the past 500 years of history, that it doesn't really have much to do with the ancient past. So the idea of constructing an idea of origins out of that, in terms of an idea of some kind of biological determinist way, is really kind of trying to build building a castle on sand. And so that's me finished, and thank you to all these people and to any thousands more who've helped me with this. So thank you.
There we go. Thank you, Tom. We have a few questions. We have time for a few questions for Tom. Anybody at all? We have a question from John Cleary here at the back.
Thanks, Tom, for a very interesting talk. I'm going to ask you specifically about the ancient DNA. If someone came to you and said, I've got the toe bone of Robert the Bruce, can I test this for ancient DNA and work out the Bruce's genome? What kind of advice would you give to that person?
What is their question? What do they want to find out?
They want to find out what Robert the Bruce's genome is. And I suppose if they're related to him, I guess, is the underlying question.
Okay. Well, you could definitely do that if you believe yourself to be a direct paternal ancestor of Robert the Bruce. You could definitely figure that out from sequencing that ancient genome. If you were anything other than like his direct paternal ancestor, then it would be probably impossible to figure out if you're his direct ancestor, because of the way that Ancestry works. You don't inherit DNA from all of your ancestors. You inherit DNA in chunks, and you get to a certain point where you have more ancestors than there are chunks of DNA that you inherit, and it's not very far back in time. So essentially you could be the direct it goes back to what I said about Charlemagne, and that everybody has an identical ancestors point, is that you have this point where everybody who existed is your ancestor, but you only have DNA from a very small proportion of those people. It's just by random chance who you happen to have inherited DNA from.
What kind of advice would you give that person to handle and deal with the bone they've got, the actual relic?
Well, I mean, yeah, there should be. It's weird because we can control for contamination. It's less of an issue. A lot of these ancient bones that we've looked at have been handled over sometime. In the case of Cheddar Man, 100 years, and we were able to control for that. At the same time, I don't want him licking them either. Handle them with gloves if possible. Don't handle them too much. Try not to breathe in them too much, and try to keep it as sterile as possible. But at the same time, if that's not been done, then it's not the end of the world. We've gotten stuff out of some pretty dodgy-looking bones in the past. Yeah.
On that issue or that topic of ancient DNA, what is the current situation with ancient DNA in ancient DNA analysis in the UK? Are we going to see within the next 5 years maybe 100 ancient genomes sequenced in Britain, or will that take 10 years?
It'll be thousands in the next probably 5 years, I would say.
Wow. Okay. And what do you think that's going to do in terms of changing our theories and views on the peopling of Britain?
I think essentially it's going to whittle down. It's a whittling sense in whittling down the hypotheses. So I think ideas around the Saxon migrations and the Rome migrations, how much they contributed in terms of ancestry to Britain. I think that those questions will kind of finally be resolved to some degree. And I mean, it's interesting because a lot of archaeologists who work in prehistory have gotten very grumpy about the fact that population change is back on the menu to explaining cultural changes in the past, particularly in the Neolithic and the Bronze Age. But geneticists complain about how grumpy those people get. Just wait until they have to deal with historians and slaughtering their sacred cows about population change in the past. I imagine that they're going to be even more hopping mad, particularly ones working with the ideas about Saxon migrations and things. But yeah, I think that there's just going to be a better idea of mobility in Britain, particularly in historical periods now, because that's the periods that big labs are moving into now. In the past, there was just this big study of Iberia, which found this, and that there were just people, moving into Iberia and influencing the genetics there sort of constantly through the past. So I imagine that the same thing, we'll just get a better idea of the overall diversity of populations in the past, and how much movement of populations you get, or a much more nuanced idea that isn't as guided by specifically history and to some extent archaeology as well.
So that's going to take us up into the last 2,000 years as well. And I guess that a lot of the theories that were generated by the people of the British Isles project, the theory that the Romans didn't really leave much of a genetic signature at all, the Vikings left a little bit of a genetic signature, not as much as you'd expect perhaps. Then the Anglo-Saxons seemed to be leaving genetic signatures all over the place, and then the Normans didn't really leave much of a genetic signature either. Do you think we'll have answers to those theories as well? Revise them a bit?
Yeah, it definitely will. And I mean, the sort of the people of the British Isles project, I mean, it was really brilliant in showing the genetic structure that there is in Britain, explaining what the historical processes that are responsible for that structure. So they put forward some very plausible ideas, but there were lots of other variable interpretations of what that structure could be, and there was a lot that was unexplained. Like there was a difference between, North Wales and Southern Wales, but the difference between Cornwall and Devon, and there was kind of some reference to population structure over the last few thousand years, but it could be a lot of different things contributing to that. It could be to do with migrations to particular regions and not to others. It could be to do with population structure as well. And yeah, the part of what people are trying to do with the ancient DNA is resolve where that structure comes from, because it doesn't necessarily all date to the same period. You could have one that's more recent than another, and you have this idea whether, so you get England has this particular, or particularly Southern England has this particular, and Eastern England has this particular signature that has been associated with the influence of the Saxon migrations, but it could just as easily be the Vikings, potentially based on the timings, or it could be, potentially more recent than that. So refining that timing is what ancient DNA will be able to do, and for sure get a complete picture of specifically population history in Britain. Then we can look at cultural change and try and see whether it's associated with that as well.
And do you think whole genome sequencing is going to make a big difference in that regard?
It does, in the sense that you get an idea of essentially what you're doing when you're doing whole genome sequencing in terms of ancestry is you're getting a sample of a person's ancestors. So that gives you a lot of statistical power in terms of inferring population change. So a lot of these studies are based on what looks like a very small number of samples, but what they're doing when they're analysing whole genome information is treating an individual as essentially a random sample of a population of ancestors. So they're looking at populations of potentially hundreds of people when they're making these inferences. So yeah, that's where it comes in. I mean, the real sort of high coverage whole genome stuff is going to be more useful for very fine scale genetic differences between populations. So looking at population movements between two places where people occupying those areas are very genetically similar to each other. So that is one of the biggest benefits. But also the big thing is going to be looking at functional variants linked to things like disease resistance, which you can only really do with whole genomes, and dietary adaptations like lactase persistence. Because the whole story of, it's a different talk, but the whole story of lactase persistence has turned out to be completely different to what people thought it was, as well as the skin pigmentation characteristics that I discussed with regarding Cheddar Man, yeah.
And will you be getting any epigenetic information out of these ancient genomes, or is that much more difficult?
It's a lot more difficult, but you can. So, epigenetics is a very broad, is a term that is used to describe a very broad range of phenomena, some of the effects of which are sort of disputed to some extent. But yeah, if you talk about methylation, which is one of the mechanisms that's described as epigenetics, so some work has already been done on methylation in Neanderthals. So yeah, that is something that we'll look into. It's very difficult to do. But it is potentially something people will do in the future if there's a reason for doing it. Yeah, they can think of a good reason.
So there's a lot to keep you busy anyway.
Yeah, loads. Yeah, and I'm just an archaeologist, not even a geneticist.
Any questions at all? Yes, we have a few here in the front row, and a question back there as well.
You mentioned about possible sampling of another 1,000 or so ancient individuals in the next 5 years, but I wonder what the picture is currently in terms of, for example, how many individuals from that sort of period have had their Y line analysed, what our current understanding is based on.
You're talking about how deep it is. So they all, to some extent, they all have their maternal and paternal DNA analysed. It's just kind of a matter of course, and particularly the maternal DNA, because you have 1,000 more copies of your maternal, of the mitochondrial DNA in a cell, that always comes out well anyway. So we always get some knowledge of the haplogroup, but it only tends to get sequenced to a particular level. So you could start getting people looking deeper into it, certainly in that respect. But the problem is that, so what I was going to say about Cheddar Man's Y chromosome, the paternal and maternal lines for the kind of studies that have already been done are less important or can be misleading because, for instance, Cheddar Man's I2A2 haplogroup, when you get that big change in the Neolithic, you get a huge change in the autosomal DNA, but the farmers that have arrived in Britain are also I2A2. So if you were looking just at the Y chromosome, you would see no change, even though there is a huge change. So this is kind of why, even though they have been done, they haven't been looked at so much in terms of the questions that they're asking. The kind of whole genome information tells them a lot more than looking at these specific paternal markers. I mean, they can be useful still and are being used to answer particular questions, but the main questions targeted by these studies need to rely more on the whole genome stuff rather than the very specific markers of ancestry. But you can imagine a situation where, particularly in historical periods like we're talking about with Robert the Bruce, someone might want to know what Robert the Bruce's very specific Y haplogroup was in order to figure out if they're likely to be a direct paternal descendant, and that could potentially work.
We have another question here.
It's when I'm thinking about sampling, because presumably over time, not every culture would bury the human remains, and sometimes cremation was used. So how do we take account for the sort of missing information? We can only test the samples that are there and the remains that have been found that are in museums or found in car parks or wherever they happen to turn up, or in the from the Crossrail project. So you're getting a very biased sample in the first place. How does that affect the analysis and interpretation of the results?
It can do to some extent, although with a lot of these prehistoric studies, what they establish, I mean, it's partly accounted for to some extent by the fact that you're looking at genome-wide information, so this population of ancestors. So in order for there to have been, for instance, a persistence of a particular population that we're not seeing, for instance, if certain people were cremated, that's fine, but when that persists through time in the way that we see, you would have to basically have a situation where these populations are existing but just not interacting with each other carnally through this time. So, which is, based on the knowledge of human history, is unlikely. I mean, you do get evidence for barriers between populations and populations not necessarily mixing so much for a few hundred years, but eventually they always tend to come together. And it's the fact that with these population changes that you get a change and then that persists through time. You know, the idea that there is a hidden population persisting that is not turning up in that sort of, it becomes too incredulous that these two populations are living right next to each other and not mixing with each other at all. I just think it does happen in Britain when the people associated with Beaker cultures arrive. I think that at this point, people in the Neolithic Britain are cremating most of the dead, and I think they carry on cremating those individuals. And as you probably guess, fire isn't good for DNA, so you can't get DNA out of cremated remains. So I think that initially there is a persistence of the Neolithic population alongside this new Beaker population, but we don't see them because they're all cremating their dead and we can't get the DNA out of the dead. But then you hit this period where the Neolithic ancestry, the ancestry from the previous Neolithic, bumps up a little bit, and I think that that's when the two populations mix. But for whatever reason, that Neolithic population just has a smaller genetic legacy. And because that persists through time and to some extent up until into modern populations of Britain, then you know that change is permanent to some extent, yeah.
And we have a final question here.
Sorry.
Thanks. With looking back at the Y DNA and the mitochondrial DNA, going back to the previous question, with the population changes you've been mentioning, how much can you tell about how much is male-driven, female-driven, or driven by both sexes?
So what they tend to look at is, so the thing is that Y DNA and mitochondrial DNA can change through reasons other than the predominant people associated with the migration. So social structures and things like that can affect it. What people tend to look at is the amount of ancestry associated with a particular population on the X chromosome. Because women have 2X and men only have 1X, you can and compare it to the ancestry across the whole autosome. So if there's a lot of ancestry on the X chromosome from a particular population and it's much lower amongst the ancestry as a whole, then it would suggest that there was more of a female think that's right. So I'm trying to retain all this in my head. There's more of a female orientation, and then if it's lower, then it's more of a male. So in the Neolithic, it's gone a bit back and forth, but it looks like broadly equal male and female. With these migrations of people from the steppe, again, it's not exclusively male, but it seems to be, there's some evidence that it's male-dominated. But there's also changes in mitochondrial haplogroups and ancestry that suggest that it's not exclusively male. It's not rampaging, horny males coming in and killing all the men and taking all the women. The DNA doesn't support that interpretation. But certainly it looks like that it was male-dominated, yeah.
Well, we have to call it a day there, unfortunately. We could talk about this for another hour and a half, I'm sure, because clearly from this presentation, the future looks bright for ancient DNA. Please give Tom Booth a very, very warm round of applause. Thank you, Tom.